What is wrong with our DNA-centric approach to biology?
Well, that depends what you want to do. So, for certain kinds of things, single gene diseases and some things like that, that might be exactly the right approach. And I mean, I think fundamentally that multiple approaches can be good or less optimal depending on what you're trying to do. So, I think that if you are trying to control large scale shape and you want to solve some problems in regenerative medicine, for example, or you want to understand better what evolution is doing, then the genes are just a part of the story. And as we know from computer science, understanding your hardware is very important. But that's just the beginning of the journey, right? Understanding your hardware is step one. The next step is how do we reprogram that hardware to do what we want it to do?
Right. So DNA, in some cases, it's you can just look at that the hardware sort of analogy that we often think about in terms of biology. I think that's maybe one of the things we did sort of in the 20th century. So we went, oh, we can zoom into the molecular structure of the cell, and then so any question in biology then could be answered just by DNA and looking at that code. So what can't be explained by DNA, and what other ways should we think about understanding organisms?
Well, think about any other task where your goal is to control a complex system. So there will always be a story to tell about molecules and chemistry and things like that. But even that is not a privilege. And Dennis Noble points this out very, very well, is that the genes and the chemistry around transcription and all of those things are not a privileged level of causation. So if you really wanted to be reductionist, you wouldn't be talking about genes. You'd be talking about, I don't know, quantum foam or something like that. But you can pick your level of description, and more importantly your level of intervention, and then you can ask how well does that work out for you. So people certainly do control computer hardware, sometimes at the level of the hardware. There are people who make chips and things like this, but when you're on your laptop, you're not programming it with hot needles and magnets. You're using this beautiful interface that the system gives you for controlling it top down. You're giving it stimuli, signals that the system is primed to respond to, and controlling it that way. And in developmental biology, I think what we have is a set of multi-scale problem-solving agents. I think evolution builds these kind of multi-scale problem-solving systems, and the optimal interaction with that system is to understand what are they capable of, what are the worlds that they're navigating, what are the policies with which every level, so yes, the molecules, but also the cells, the tissues, the organs, and all the way up, what spaces are they navigating with what goals and with what competencies. That really allows you to pick the right level for what you want to do. And using that kind of approach, which pretty much is just taken from neuroscience and behavioral science, we've discovered a number of new capabilities in regenerative medicine, in cancer, in birth defects, and so on, that are much more easily reachable that way.
Right. So those other problems that you then allude to, what in controlling. So imagine we go, okay, we're not going to reduce everything down to DNA because that's, as you kind of say, an arbitrary level of analysis. Why DNA? It's important, but then why not go down all the way to molecules and the physics of it? So there's multiple ways you can see biology. So the reason why I bring up DNA right there at the start is you have some experiments where it seems like, or there are some organisms where the DNA matters almost not at all. The organism kind of completely ignores DNA, and there's some work on even and then we have like stories we tell about why we get cancer, and that's to do with mutations in DNA, and that's what causes things like cancer. But then if we change our story so it's not so DNA centric, it feels like it kind of there's an upheaval into many aspects of how we think about biology. So, what then? How do we start building frameworks? More specifically for understanding these kinds of things.
Well, let's talk about an example. Let's talk about the cancer example. I mean, the reason these frameworks are important is because they constrain or facilitate new approaches. They constrain or help you see new types of experiments that you could do that will help control the system. So let's talk about cancer. If you have a gene centric view of cancer, which by the way is crumbling for many reasons, not just our work, then you've got this fundamental focus on cell cycle checkpoints and DNA damage and things like that. And what you would say then is that you would have two predictions that which you would make. One is that animals whose bodies have ready access to huge numbers of very plastic, highly proliferative cells should have a high oncogenic cost. And this is a lot of times why people say why humans are not highly regenerative, is because we want to live a long time. And if you have all these plastic cells around, then you're likely to end up with cancer, right? So you would say that plasticity goes with cancer. And the other thing you would say is that if you do have cancer, the only thing that is left for you to do is to try to kill those cells because they're irrevocably broken, they're damaged. You got to kill them. So that's chemotherapies and things like that. But there's another way. There's another way to look at this, which is to ask the question: why is there ever anything but cancer? In other words, why do individual cells, which at some point were unicellulars, little amoebas and such, and they had their own little tiny, what I call a cognitive light cone, which is the size of the biggest goal that they can pursue. So these little tiny cell level cognitive light cones, with respect to metabolic goals and proliferative goals and things like that. Why do they get together during embryogenesis and work on these grandiose construction projects? And we can talk it's separately about how I'm using the word goal and why I think these are actual goals. But what enables them to work together is a very specific connection policy where they join together into networks. They partially give up their individuality, they share information, and they form these large scale collectives that store pattern memories of growing large things, tissues and organs and things like that, growing and then maintaining those things. And so then you might make the exact opposite prediction. You might say that, well, then animals that are really good at doing this, so highly regenerative, highly plastic creatures that are very good at exerting morphogenetic control over plastic cells, should actually have less cancer. And also, and so that turns out to be true. Like planaria and salamanders, which have a huge plasticity, are actually very cancer resistant. And then perhaps more importantly, you might think that the way to address certain kinds of cancer would not be to necessarily try to kill the cells, but to actually reconnect them to the rest of the network. In other words, to reinflate their cognitive light cones so that it's big enough to be building organs and tissues, not just proliferating and migrating. And so that, in fact, is what we've done. We've shown this in frog in the frog model injected with human oncogenes. The first thing, and this has been known for a long time, the first thing that cells do when they express an oncogene is they cut off electrical communication with their neighbors. And that shrinks that cognitive light cone, that boundary between self and world shrinks back down to the level of a single cell. And so they don't become more selfish, it's just their self is smaller. And their actual self shrinks, and the scale of their goals shrink, and you get individual cells that treat the rest of the body as external environment. So what you can do is you can control the bioelectrics, ignore the DNA damage entirely, and you get cells that contribute to normal skin, normal other body organs, and so on. And so that's just an example of asking these kind of questions, which at first seem pretty weird. We're talking about goals, we're talking about the boundary between self and world. These are very sort of psychological kinds of things, but all of that stuff in our brain and our nervous system didn't arise out of thin air. It evolved from systems that were already doing this kind of thing to navigate anatomical space for a really long time. And so that's what this enables you to do, right? Going beyond a privileged level of causation, such as chemistry, lets you think about these other ways to understand your system.
So, and you bring up this. So one way you could do that is through bioelectricity, and yeah, it's weird because that's a kind of a question I had more generally about biology, is why, if we then think about, we have however many cells in our body and then they all decide to work together, but then in cancer we have this scenario where seemingly some are going rogue and deciding, actually no, I'm not involved in this collective anymore, I'm doing my own thing. And so, for example, do we know that in our own bodies, can we see, is there something to do with bioelectricity? Is there, like, what else is going on then? Is it a different story for humans versus other organisms? But like, is there even a way we could think about ourselves and bioelectricity going wrong? It's because it's weird why, like, we now need to start thinking about what causes some organisms and some collectives to start saying we're going to be in a network and others not to be, right? And what decides that?
Yeah. So there's two questions here. The first you asked is how much does this apply to humans? Yeah, I mean, all of this stuff is incredibly highly conserved. We see ion channels as oncogenes in human cases. We see it in mice. There's absolutely no reason why this would be different for humans. I don't know yet how much of clinical human cancer is traced to fundamentally a bioelectric breakdown. Some of it might be stress related. You know, cells are more likely to leave a collective when it's not working out well for them, when there's high stress. But we've certainly been able to induce a kind of, for example, metastatic melanoma in frog with no DNA damage, no oncogenes, no carcinogens, just a disruption of electrical communication between cells. So you certainly can cause that way. How much of that is clinically what happens in humans, I don't know, but all of the pathways are conserved and they're basically the same. And so I don't anticipate there's going to be that much difference. Now, the other thing you asked, which is why do some cells do it and some don't, is kind of the bigger issue around understanding the policies under which cells join into larger groups. And some of that is evolutionarily determined in the sense that there are specific mechanisms that induce cells that connect together with their neighbors. They throw up gap junctions. They start connecting in these larger scale networks and so on. Some of it is it's not directly evolutionarily selected for, but it's provided because the hardware has these kind of properties that allow it to solve problems on the fly. So we've seen through, I mean, well, developmental biologists have had these examples for a really long time of doing these very strange things to embryos that have never happened directly naturally during evolution, and yet they can handle all of these things. And that's, I think, because biological tissue has this amazing property of being able to find different ways to navigate anatomical spaces, physiological spaces, transcriptional spaces, and that ends up cashing out in terms of you're put in a completely novel scenario, but you can find your way to a coherent way of being an embryo or another, some other kind of construct. And so we've seen that in our synthetic things that we make, xenobots and anthrobots and other things. Yeah, and developmental biology has seen lots of examples of this.
Interesting. I mean, there when you talk about these generalizable skills, so that you throw some problem at something and it doesn't necessarily need to be hard coded in DNA, but somehow these biological materials have this kind of capacity to deal with that change. And then why do they have that capacity? There's some kind of goal stored there, but and you said, well, maybe that is stored in the network somehow overall. There's a kind of pattern there, and that's kind of what I wanted to ask about next is where are these things stored? How should we think about the nature of memory? Something I was listening to you recently talk about butterflies, and when they change, they grow from they go caterpillars turn into them, and yet somehow there's actually a way in which they have some kind of learning that they retain as they go into butterflies, and yet it also that learning changes obviously because they're now a butterfly, they're completely different. So how where is that stored if they're completely changing, and there's kind of loads of examples. You also have, you cut the heads off these kind of worms, and yet so when you tell me a bit about memory and where these kind of patterns are stored.
Yeah, yeah. There's a lot there. Let's see if I can remember all the things you asked. So the first thing is, where are these patterns stored? So within a given animal, we can see, at least in the cases that we've studied, we can see the pattern. We know where it's stored. You can we have now techniques to visualize a memory in non-neural tissue, and by using these voltage reporting kind of techniques, and you can actually see the pattern in the tissue that serves as the scaffold towards which the cells will build. So we can kind of like neuroscientists do this neural decoding, right, to try to extract the cognitive content of a brain. We can do that with a morphogenetic collective intelligence and read out the patterns that it's trying to implement. So that we can see. Now there's a deeper question, which is, where do these patterns come from in the first place? And I think there are a few things. I think that's an incredibly deep question. There's a few things that we need to change in order to try to answer those questions. The first is this idea of, where do patterns come from? So there's a notion of emergence that's been around for a long time, and so things like the nice bell curve that you get when you dump a bunch of marbles through a Galton board, or the hexagonal cells of a particular size. When you heat up water on a pan, you heat it evenly, you get these Bernard cells, right? There are all these kind of patterns, and the question of where does it come from is not going to be answered by looking at the physical hardware of the system and say, oh look, I see the encoding. It's written down, like there's a little picture of the Bernard cells in the bottom of the pan. Like that's not where it's going to come from. And so what I think is happening is that evolution, by building bodies, evolution is basically building pointers into a huge Platonic space of affordances, like the laws of computation, the laws of mathematics, adhesion and the bioelectrical signaling and all these things. I think that's what evolution is doing. Evolution is building physical machines that couple to these things that live in the space that people have been talking about since the start of Plato and Pythagoras talked about these things. So I think that's probably what's happening. So there's a combination of the physical hardware, which is shaped by evolution to be compatible with and make use of these specific, you might call them free lunches how physicists refer to them. So that's part of it. Part of it comes from wherever it is that mathematics comes from. So the same, the truths of number theory, the different facts about different shapes and so on. That isn't physical. That isn't anything you have to evolve. It isn't anything that's determined by the facts of the physical universe. It's just there somewhere. So part of the patterns come from there, and part of the pattern comes from the emergence of the physiological software working through the hardware machine, kind of like Turing patterns and things like that, right? So those are some of the factors. Now, what evolution is apparently very good at is producing hardware that, by default, with nothing else happening, is very reliably will reach a particular point in anatomical and physiological space. So we get lulled into this false sense of security that that's what the hardware encodes, but that's actually just the default outcome. And what you find out is that if you perturb the system, it's actually capable of many things. So I think beyond emergent complexity and beyond emergent patterns, what you actually have is emergent goal-directedness. And I think that some of the forms in the space are not passive forms like hexagons and things like that, but actually ways of problem solving, ways of navigation. More globally, if you want to kind of go deeper, I think they're ways of sense making, and I think there are ways by which any system can build an internal representation of its environment and try to minimize and maximize certain things that it likes. This feeds into the second thing you asked for, which is the nature of memory. And what's interesting about the butterfly and caterpillar case, just to kind of lay out the facts here. So you take a caterpillar, you train it to eat leaves on a particular colored disc. The thing metamorphoses, completely refactors its brain, builds a totally new body that's controlled a very different way, lives in a three-dimensional world. Now it flies around, doesn't want leaves, it wants nectar. And yet the butterfly has you can recover those behavioral memories. And so yes, there's an interesting question of where does the memory persist during the brain, the destruction of most of the brain cells and then a rebuilding, where is it? But I think even a more interesting question, which is what you alluded to, is the fact that you can't just store that information the way we do in our computer system. Storing the memories of how you move a caterpillar body to eat leaves is of no use to a butterfly. So what's happening here is a remapping of the information onto a new form, where what is propagated are not the details of the memory, but the salience. So it doesn't. I think, and this is just something very recently that I've started focusing on, is this idea that biology isn't committed to fidelity of information; it's committed to saliency. And what that means is that at any given moment, and let's just think about how it works out for us as opposed to caterpillars, butterflies. At any given moment, you don't have access to your past. What you have access to are the engrams, the traces that the past has left in your brain and body, and you have to continuously interpret them. You have to tell a coherent story. In humans, that's linguistic. In other animals, it's probably not. But the idea is that you have to constantly interpret your own memory traces to build up a coherent story of the world and of yourself. And that fact, the fact that memories are basically messages from your past self, and like any message, they're subject to interpretation. You don't have to take them as seriously, or you don't have to interpret them the same way that your past did. And I think where that comes from is evolution, and the idea that everything is going to change. Not only is the environment going to change, but your own parts are going to change. You're going to be mutated. There's going to be all kinds of things going on. You can't overtrain on your priors, as the machine learning guys would say. It's too it's not a good strategy to assume that the future is going to be just like the past. So I think what happens in biology is that it's a kind of bow tie like thing that again is used in AI. It's used for these kind of architectures where you have these large layers and the information comes in, and then it's an auto, they call it an autoencoder, and in the middle, the layer in the middle is quite small. And so what happens is, in order for that autoencoder to work, you can't try to memorize the inputs. You can't just study your data set, because that middle layer is tiny. You're not going to get through. You're not going to get all that data through there. So what that system has to do is generalize. It has to abstract from the particulars of your experience down into a memory that's not a memory of the specifics. It's a memory of some compressed, highly compressed representation of what happened before. Now that means that the right side of that funnel, right, that bow tie, the right side has to be creative in interpreting what this is, because these are not direct memories that it can use directly. This is some kind of compressed model that it has to then reinflate, reinterpret, and there is no metadata about how this was interpreted before. It's up to you. You interpret it as best as you can in whatever adaptive way is useful for you. And in particular, the better that works and the more compressed it is, things that are data that's maximally compressed starts to look very random. This is something that people point out in the SETI world, the search for extraterrestrial intelligence, that if you have a very sophisticated signal that has huge compression, it looks random. And so that means that the right side of that bow tie has to be really smart. I mean, the left side has to be smart in generalizing from particulars to a general kind of some kind of compressed representation. The right side has to be it has to be creative in reinflating and decompressing that message into whatever is salient for it now. So I think that goes on everywhere. I think it happens at every level of biology. You can look at science that way too. You do all this work and then you compress all your wisdom down into a paper, which is this like really sparse, especially like if it's a Nature paper or something. It's this crazy sparse thing, and then other people will read it and try to reinflate it into brain and mind states of their own, which may be nothing like yours. But again, it's that bow tie, like it's everywhere. Language is the middle part of the bow tie for us, and so on. And so I think that's what's happening with information, and that's what happens in evolution, and that's what happens in development, is that you get past this egg. You don't get past the whole body that you then try to scale up. You get past an egg, and the system has to interpret itself in what does that egg mean. And under normal circumstances, a frog egg makes a frog, and a acorn makes an oak tree. But actually, if you have the oak genome, and a little wasp comes along and sits on that leaf and gives certain signals to it, what it ends up building is this crazy, beautiful gall, which is this like spiky red and yellow thing that you would have never in a million years thought that those leaves are capable of. And so again, right, reinterpreting that same genetic information that you have can give you very different outcomes.
With when then, so organisms kind of they kind of must have this generalizing ability, because as you say, they're constantly taking damage. They're changing, even though I remain the same in terms of my form and structure. All my cells are changing all the time. And then I'm wondering, how does evolution select for these kinds of things? If it's like, at least on one level, we think about it as it needs to at least survive to the next generation. So how should we think about then the process of evolution itself? Is it just the case that I like that? Yeah, I like that you talked about. There's almost an analogy between different spaces. So the way DNA transcriptional space to morphogenetic space to bioelectrics. Maybe actually that is analogous to moving in 3D space, and so there's not necessarily completely new things you have to learn. So, but then yeah.
I think that's right. That's exactly how I think about this. Is that evolution pivots some of the same tricks. So first you had some kind of metabolic space very early on, and then you had physiological space, and then genes came along, and you had transcriptional space, and then multicellularity came along, and you had anatomical morphospace, and then brains and muscles developed, and you could move in three-dimensional space. And so that's our conventional behavior. But all of these spaces were used before then, and now we have linguistic space that you can live in. And so I think there are all these spaces, and even not only I think are the policies for how to navigate them conserved, but even the molecular mechanisms. I mean, the reason that brains and neurons are so good at doing what they do is because they're just reusing the same machinery that cells were using to navigate anatomical space long before they appeared. It's just there's a couple of interesting pivots from, for example, from spatial reasoning and development to temporal reasoning and behavior, right? So you're now more paying attention to time versus space, but otherwise same kinds of things. And so, people do talk about the evolvability and where evolvability comes from. My gut feeling is that it probably predates life. So I think this kind of intelligence and problem solving is a bigger set than living things. And I think that even at the very beginning, I mean, we've shown that if you just have a system of coupled differential equations, which, for example, describe gene regulatory networks, but they can also describe a million other processes, that those kinds of systems are trainable. They have 6 different kinds of memory, including Pavlovian conditioning. You don't need a cell. You don't need biology, right? It just falls out of the maths somehow. And so that's what I think it's doing. I think evolution is capitalizing on this problem-solving competency that is baked in right into the fabric of the universe, I think.
Right. Well, so the final thing, let's talk a little bit about that, because on you have like computer programs that these algorithms that are very simple, but there's a way in which they actually they're an algorithm, so they're going to get to the endpoint anyway, but there's some interesting things that come along the way that they can navigate problems in a way, and we kind of anthropomorphizing it, and maybe it kind of ties into the way you talk about Platonic forms, and so
Yeah.
it's even before life in a way, because there's just something inherent about the structure of our universe, our reality, that actually is able to solve problems. So why don't you? It might be, you don't need to go in detail to maybe explain how that works. Maybe even the principles. What does that say then about us?
Yeah, I think a few things. First of all, I don't think there is any such thing as anthropomorphizing. I think that's an old term left over from when we thought that humans were magical, and it was kind of bad to attribute that certain properties to things that aren't humans. I think that's a pre-scientific notion, so I don't think that exists. That's the first thing. The second thing is what we see in these algorithms. What I try to do is to really hammer this point that. There is not just emergent complexity, but emergent goal-directedness, which means that when you make systems, just because you know the parts that go in them, it doesn't mean you know what the system is going to try to do. I mean, some engineers will say that's obvious for complex systems in artificial life. You often get surprised, you get perverse instantiation, you get all these things where you were surprised. But it's still the case that a lot of people feel that they can look at something and tell you how intelligent it is. So people will say, oh, well, that's just a collection of genes. That can't, that's just a chemical system. That's not intelligent. And I really strongly think that our intuitions are terrible in this area. We are badly calibrated. We don't know. And more generically, you can't decide how intelligent something is from an armchair. You have to do experiments, perturbative experiments. Observation is not enough. You have to actually make hypotheses and test them about the competency of that system. So I try to kind of nail this concept by finding something extremely simple. And this is the work of Taining Zhang, the student that did this with me. What we said is, okay, let's take something that everybody thinks they know, and these are sorting algorithms. So the kinds of things that all computer science students study these things for decades, right? So bubble sort, selection sort, that kind of thing. Very simple algorithm, 6 lines of code, completely deterministic. There is no new biology to, the thing about it, doing this in living systems is that if you see them doing something amazing, somebody will say, well, there's a specific mechanism for it, you just haven't found it yet. Okay, so here's a system where there are no more details to discover. We know exactly how it works. We know every piece that's in here. And so by treating their journey in sorting space, right? So they sort, it's an algorithm that sorts an array of numbers. We did some tricks that I don't have time to tell you about now, where you can basically look at this as the journey of an agent through sorting space. And then we ask some simple questions about what competencies does it have to undertake that journey, because you think you know what it's going to do based on the algorithm, and it turns out that that they have some very non-obvious behavior, including delayed gratification, so the ability to actually unsort the array if it's going to help them later on, which none of that is in the algorithm. And they also have the ability to once you parallelize it and you make each individual cell able to sort itself, you find out that they have a functional ability to recognize other cells of the same algo type, meaning of the ones that are running the same algorithm, which again is nowhere in the algorithm to recognize anybody else doing what you're doing. So these things, and so my point is simply that if demo bubble sort can do these things, how much more emergent problem solving are we going to find in complex systems that we build, including language models and various kinds of AIs. And now there's people doing these minimal active matter, droplets that have 2 or 3 chemicals, and they can do interesting things. So I think the message is to have some humility here and understand that our ability to recognize intelligence is not very good, because we can't visualize these other spaces, and because we have this hyper focus on brains and neurons and things that look like us.
Yeah, and do you think beyond, above humans, so like looking at really small things, really simple things, it actually kind of surprises us. Like, do you think you can measure the competency of economies of weather patterns, and more interestingly, do you think that they might be more complex, more interesting than we are, but we just can't understand, like, their morphospace? We don't actually just understand the kinds of things they're doing.
Yeah. Well, two quick things. One is that while there is no guarantee that larger systems will be more cognitive than their counterparts, and I don't think there's any guarantee of that, but it's entirely possible. And I think that both the kind of profligate animism, where people say there's a spirit under every rock, and the ecosystem is its own intelligence, and the planet isn't, like, you can have those hypotheses, and that's great. But saying that is just as bad as the teleophobia that scientists have, where they say impossible, those things can't, whatever, they just can't. It's a category error. I think both of those things are wrong. I think the right approach is to do experiments, to have hypotheses and do experiments. And so yeah, even though it's difficult, expensive, possibly, in some cases impractical or unethical, but when people say to me, oh, well, you might as well say the weather is alive, and I say, well, I wouldn't just say it's alive, but if you wanted to come up with a way of trying to see if a hurricane has habituation, sensitization, anticipation, associative learning, I don't think you could tell me that that it can't. I think you'd have to do the experiment. And I think that it's entirely possible. And we have some experiments going on training ecological simulations like Lotka-Volterra equations and things like that. There are a lot of surprises to be found, but you can't just declare it. You have to, you have to do the experiments.
Awesome. Thank you very much. And that concludes the podcast. Thank you very much for listening. And if you want to check out similarly awesome guests as Michael, then click on one of these videos, or check out some links in the description of people in Michael's lab where we discuss similarly cool topics. Have a good day, guys. Thank you.